everyone, welcome back to this cell signaling course. If you recollect, we were discussing how two-component system is working in microbial world. We took example from bacteria and fungi and saw how different processes like sporulation, quorum sensing, biofilm formation, chemotaxis or life form equivalent to asymmetric cell division was being regulated by two-component system. What we have discussed earlier that signaling occurs in all different life forms, and that too by different mechanisms. So we took example of two-component system as one of the simplest type of signaling event where a receptor is involved, signal is involved, and there are effector molecules which are involved. If we talk about plants, those are unique in terms of possessing both two-component signaling and receptor serine threonine kinase signaling. In one of the earlier classes, I emphasized that animal system generally lacks any of these receptor histidine kinase signaling, which is key in the two-component signaling. What I also emphasized that plants do use both two-component system and receptor serine threonine kinase system for signaling. However, plants clearly lack receptor tyrosine kinase signaling. So in today's class, we will see how two-component signaling affects or it is arranged in plant system to mediate the responses. So we understand that plants are complex and they are largely sessile organism. When I say plants are complex, just not because of their magnanimity of their size, but also because we know that the ploidy level changes which are afforded by plants is way more than what animals can afford. We understand that plants do need nutrient, all such factors so that they can grow, whether it is abiotic or biotic factors. And to respond to those factors and so on, plants also utilize hormones, and we know as many as 5 distinct hormones which are present in plants. And these hormones help in the process of growth and the homeostasis which is there taking place in the plants. One of the most important hormones which we will study in context of two-component signaling is cytokinin, which is responsible for growth and it regulates the cell division. It is normally synthesized in the roots, and using the xylem, it is sent in the upper parts of the plant. As well as in combination with auxin, the other most important type of plant hormone, cytokinin induces root development or shoot development. What is understood is that low cytokinin level induces root development and high cytokinin level promotes the shoot development. Another plant hormone which we will study in context of two-component system is ethylene, which is responsible for fruit ripening. We will also see that how two-component system is involved in the ethylene signaling, it is involved in the cytokinin signaling, thus affecting the root and shoot development, as well as it is important player in phytochrome signaling. So normally, if you know these two-component system which are present in plants, they pose a unique opportunity for researchers to understand them and discuss them, because mostly we have hybrid two-component system in the plants. If you recollect, hybrid two-component system automatically suggests that your receptor had the histidine, where initial phosphorylation utilizing ATP happens, and the same receptor also has a conserved aspartate where the phosphor transfer from the histidine can happen. There are also examples of divergence in the two-component system. We will discuss more about that, but just to initiate you here, the divergence refers to the different types of receptors which are present which are non-canonical. Non-canonical here would mean that Suppose you are talking about the receptor and hybrid type of receptor, so you are expecting the histidine to be present. But there are examples in plants where the receptor lacks the histidine. Similarly, there are response regulators which lack the aspartate residue. So they pose unique challenge in signaling, particularly two-component system in plants. As I mentioned earlier, primarily in plants you have TCS, which is involved in cytokinin and ethylene signaling, and as well as phytochrome and the function of circadian rhythm. So let's check back again and see what was two-component system. As we saw in case of bacterium and so on, you will need the receptor kinase, which is histidine kinase. We saw in case of bacteria it was called sensor kinase. I'm not sure if you call them that with that name in plants, rather they are just the kinase, histidine kinase, as often they are referred. And since we are talking about a hybrid type of two-component system, we are talking about the hybrid type of receptor, where you will have the ligand binding domain, which will be normally projected outward, outside the cell. Then you have dimerization and histidine phosphorylation domain, where basically this catalytic activity, the kinase activity, utilizing ATP, it will transfer the gamma phosphate to the histidine residue, and this histidine phosphorylation event will trigger that phosphotransfer to the aspartate residue, which is still part of the receptor itself. As the signaling moves forward, you will have additional molecule required to ensure that signaling is moving in right direction, because we understood that the movement of phosphate group in two-component system follows this route of histidine to aspartate and aspartate to histidine, and then further histidine to aspartate. So this histidine phosphotransferase molecule which is required here, often they carry the minimal domain with conserved histidine. Now they basically serve as a relay center, and they send the phosphate from their histidine to the aspartate residue, which is present in the receiver domain of the response regulator. In case of bacteria, we saw there could be multiple different types of response regulator. Here the first case where output 1 is written, it represents a scenario where the minimal domain is present which has the conserved aspartate residue, where phosphorylation can happen. Additional type of response regulator can have a new domain in addition to the receiver domain where phosphorylation will happen. And this we have multiple times discussed, that this new domain is going to define what is going to be the response. We saw it could be a transcription factor, we saw it could be an enzyme, we saw it could be a molecule which induce an interaction. So when we talk about the two-component system, I also mentioned that there are divergent type of components in the two-component system of the plants. So let's take some example, or what kind of diversity or variance exists in case of plant two-component system. I told you that there are two basic hormones to which the plants respond through the TCS signaling. So if we think about cytokinin alone, and we talk about the receptor, there are multiple genes which code for receptors for cytokinin, and they are present in different tissues in plant in different forms. If you also recollect my previous discussion on the fact that I relate the cell to a smartphone, where different apps ensure that cell is able to do the job which it is destined to do. Now you think about a scenario where a plant cell which is part of leaf versus the plant cell which is part of the root, or the plant cell which is part of the flower or developing embryo, right? They have very distinct function to perform. So, and probably all of them will be responding to the cytokinin. So you have distinct type of receptors present in those regions. As you would expect, any of these receptors will have extracellular domain, then you will have the kinase domain, and then you will have the receiver domain. Why receiver domain? Because this is a hybrid type receptor. Lot of studies have been performed in Arabidopsis, from where people know that multiple different types of receptor for cytokinin do exist. So cytokinin is a small molecule. I told you it is synthesized in plant roots, and it is derivatized from adenine. So it's a small molecule which is sensed by this extracellular domain, which has this ligand binding activity. There are multiple of these which have been identified. Also, we will see as we move further in the classes that there are meristematic tissues which are unique to many of the plants. There you will have a very unique requirement of these receptors. So a very specific type of receptor will be present in the meristematic region, which will not be present in the normal somatic tissues. So in addition to these structural sequences which are present in addition to these domains which are present in the receptor, there are example of plant cytokinin receptor where there is additional domain which is called receiver-like domain. What receiver-like domain could mean? That you have a additional region which looks very similar to receiver domain, but would confuse the signaling by virtue of its presence, or it may divert the signaling into a different direction by virtue of its presence. This will definitely have its own histidine residues. There could be receptors which actually lack this histidine residue, and they will be having an arrangement where you can have RLD or without RLD, with or without RLD. You can have these receptors where this histidine residue is missing. So what you can understand, that it has the kinase domain, it has the receiver domain, but it lacks the histidine. And you remember, the signaling starts by first phosphorylation event of the histidine residue. So this will be like a pseudokinase. And in one of the previous classes I mentioned that there are a lot of pseudogenes which are involved in the signaling, and they regulate or divert the signaling from desired direction to somewhere else. And such a receptor where you lack this histidine is part of the phytochrome signaling. So you have the pseudokinase kind of arrangement where the desired histidine is missing. So not only for cytokinin but also for your ethylene, you have the receptors. So what you would notice that there are ethylene receptors also which have similar arrangement where you will have ethylene, and this is also a hybrid kind. So what will happen here? Remember that ethylene is a gas. It can actually pass through the membrane, so it will come inside, and you will find such receptors on the surface as well as inside the cell, somewhere in the vicinity of nucleus, so that they can regulate the process. So here they have this domain. If you recollect, we discussed this in case of bacteria, JAF domain. This will help it sense the presence of this ethylene-like moiety and pass the signaling on. Now if you think about the other components of the signaling in plant, you will have this HPT, histidine phosphotransferase. You will expect it to have the histidine considered residue. Sometimes you also come across this nomenclature in plants that there is an authentic kinase or authentic receptor versus a variant receptor. Remember that variant is because it may lack the histidine residue. Similarly, for HPT or histidine phosphotransferase, you may have a variant where histidine is not present. So this variation is coming to regulate and give another dimension to the signaling which was started at the cell surface. As well as you will have these response regulators. Remember, response regulators can come in different flavor, but they must have at least one domain which is like a receiver domain, which should receive the phosphate group from this histidine phosphotransferase. In addition to that, they can have domains which will divert their signaling. You can find examples where you have DNA binding domain, as well as there is something called activation domain, which makes them a suitable transcription factor. You can also have different motif which will engage in protein-protein interaction, as well as they might affect during the morphogenesis processes, they might affect the cytoskeletal arrangement. And as we saw in case of kinase, there was a variant. Here also you will find a variant which will be pseudo response regulator kind of arrangement, where this receiver domain does not have the aspartate residue, rather it is either changed to asparagine or glutamate. So you will find the problems because of which this will not be serving as a canonical response regulator. This is how domain and its variants will be present in the plant system. Let us have another look and see what kind of response regulators are present in plants. And as I told you a little early that Arabidopsis has been one of the model organisms which is studied, but we know that there are a lot of crop plants which are used, and most of them have been studied for two component system, but not as great in detail as in case of Arabidopsis. So there are histidine kinases, which are present, which will ensure that you have the domain arrangement of transmembrane domain, then you have the kinase domain, then you have the receiver domain. And we saw there could be variants also. So they have been studied in Arabidopsis, tomato, and other useful crop plants. So you have histidine kinase, similar arrangement over there, as well as the variant in them. Then you have the histidine phosphotransferase. They have been studied in maize plant also. I was telling you maize has been another important plant which have been studied in greater detail. Then you will have the response regulators. And when I say domain arrangement, we saw previously that if there is a minimal receiver domain, in addition to that you can have multiple domain. What you notice is that in case of receptor there was a receiver like domain. There is a possibility that you have the response regulator with receiver-like domain and regulating the functions, and they have been studied in maize, Arabidopsis, tomato, and so on. There are multiple different types of response regulator in the plants, and they have been categorized in two types, type A and type B. Type A are generally smaller ones, and the type B is bigger one. Automatically it suggests that there are more additional domains in type B type of response regulators than in type A response regulator. By virtue of their size, they have different domains, and that's how they direct the signaling in a given direction. Now if you look about the domain arrangement, more so in terms of understanding that how they will function, you will realize that if you take example of the cytokinin response receptor kinase, it has obviously an N-terminal domain, and then it has the histidine kinase domain, then additional domain, and then finally you have the receiver domain. You can also have for ethylene response similar domain arrangement. Remember, depending on what they are responding, this extracellular domain will be of different type. You remember in case of bacteria we saw JAF type, PAS type, HAM type domains. They have distinct property so that they can bind to different types of molecule. Then there are response regulator. If you look at the arrangement of their domains, there could be a response regulator which has the minimal domain, or there could be a response regulator which has many more domains and features. When we say domains and features, like domain which provides its activity, we saw example of transcription factor activity, we saw example of catalytic activity, and when we say features, there could be feature which is like, sometimes you would have studied this something called nuclear localization signal. So it makes the molecule move inside the nucleus. Then there could be the domains for HPTs, and we have understood that it should have the histidine residue for phosphotransfer type of event. Now if you think about the combination of these receptors coming together and signaling, remember we took an example where a simpler or canonical two-component system was giving input to a hybrid type of two-component system when we discussed the Caulobacter crescentus, div-J, div-K, and CCKA and CTRA. So many types of cytokinin receptor or ethylene receptors are present in case of plant. So many histidine phosphotransferase molecules are present, and different response regulators are present. So do they crosstalk with each other freely, or there are some tight regulation? Remember that the presence and their relative concentration is going to be very important. You can have 3-4 types of histidine phosphotransferase in the genome, but a particular cell type might be expressing in good amounts only one of them or two of them, and they will be engaging in signaling. So let's take some example here. If you think about the different kinases, we're talking about CRE1 or the other kinase which is CKI1. These are all cytokinin responding type of receptor, and then there is a ETR1 which is responding to ethylene. I am not drawing subdomains because we have understood quite well now that minimum 3 domains we are looking at here: the extracellular signal receiving domain, the kinase domain, and then the receiver-like domain. Now if you think about the type of HPTs, histidine phosphotransferase, if you take example of 2 types which are there, and they are called AHP1, AHP2. And then you have response regulators. Remember, type A and type B. Type B were generally bigger than that. So is it like all possible combinations exist here? No. So you have situation like this. You have reactions like this and so on. Okay, and then you will have reactions like this, then reaction goes this way and reaction goes this way. So there is lot of interconnection, crosstalk exists, but it is not like that all of them are interacting with all other. Like here you see HP2 does not seem to interact with the type A of response regulator. So we will see in coming classes that how the combination is going to be important. One of them will be expressed, one of them will not be expressed, and that will drive the signaling in given direction. We will see more of that as and when we move forward into the signaling course. But remember what we have understood, that same as bacteria, there will be a receptor on cell surface. If I say it is cytokinin as the receptor, by virtue of being a hybrid kinase, it will phosphorylate itself on a histidine residue, pass this on to the aspartate residue, engage with HPT, and then divert from there to the response regulator. Response regulator 1, response regulator 2, they may be interrelated or they could be independent. Okay. So in summary, what we have understood in this class is that two component system is very well utilized in plants. In addition to regular canonical and hybrid type of two component system, you do find variations in the components itself, which will have another level of regulation on two component signaling in plants. Cytokinin and ethylene, the plant hormones, they are the ones which most prominently respond through these two component system. We will see that how the cytokinin signal also responds through some of these serine threonine kinase receptors. Okay, and then this signaling helps plant perform the growth and development kind of activity, and also we will see that how it will affect plants in their stress response properties. Thank you.